Testing, testing, testing. Start. All right. Good evening, everyone. Pursuant to the Chapter 2 of the Acts of 2025, this public meeting will be conducted via remote participation. A recording of this meeting will be made available along with the minutes on our website, somervillema.gov/biosafety. I just want to say thanks to everyone for attending tonight and committing your time to this important work and transparency for a exciting and vibrant and new industry to the city of Somerville. So I really appreciate you all attending. We have a busy schedule ahead of us today, so we're going to jump right in with roll call. So I'm going to go ahead with that roll call. Just a simple aye is sufficient. Karen Carroll. Do not see Karen. Elise Belarge.
Aye.
Rachel Nadkarni. Alex Vera.
Aye.
Deputy Chief Bill Hallahan.
Here.
Captain Tim Bakey. I think Tim is here, isn't he?
Yeah, I think he was having trouble with audio.
Okay, all right. We'll check him in maybe later. Francine Rogers.
Aye.
William Fisher. And Colin Ziegler. And with that, we have met quorum, and we're going to go ahead with the meeting with the first item today, which is actually the introduction of William Fisher, the Director for the Office of Emergency Management. Bill, if you want to go ahead and introduce yourself to the group and a little bit about your work at OEM and your contribution to the committee.
So again, it's actually Bill. I go by Bill. So Bill Fisher. Been with the city for, actually finally came around to about a year now, working in the Office of Emergency Management, and the deputy Warren came on board about 6 months ago. Previous to working for the city of Somerville, I worked for a number of years for the Massachusetts Emergency Management Agency as a local coordinator in the eastern side of the state, working with communities on emergency planning and response and that sort of thing. And so, getting to Somerville, we try to assess what we had going as far as structure that we need to have in place. Same thing I would do when I went out to a town and I assisted them and try to build it up. But we're a two-person department. It's a kind of a big, lofty goal. But one of the things is around making sure that we have proper planning and collaboration for hazardous materials and that sort of thing. So we established the Somerville Local Emergency Planning Committee. So that's been established again, as well as being a member of Mystic. And this, when Colin reached out, of course, I said yes, because this folds right into that. It's a big piece of the general overall emergency planning. And any opportunity to collaborate with colleagues, internally and externally, is obviously a win for everybody. So glad to be here, and that's just about me. Looking muted.
Colin, you're muted.
You're muted.
Oh, I was going into a whole tangent about how exciting it is for Bill to be serving on the committee to provide his perspective, how it's important that this committee, unlike others, is very staff heavy. It's important for the city to be involved in these types of things and create those connective bridges, but also community members at the same time. So it's this nice blend that we have here. Looking forward to having Bill serve as well. Great. So I'd like to do a quick motion to move to the next item, which is going to be the presentation of the applications. Some quick housekeeping on how this is going to work. We currently have Don Crooks here to present on the applications for some specific contents of them because they are in the same building. They fall under the same company as subsidiaries. So he'll be presenting on the facility, the description of the health and safety training, the permits and the timeline, and then the contracts that they have in place that sort of span across these applications. And committee members who have been able to review these will notice there are similarities across each application. So Don will be here to present on that initially. From then, we'll have each of the applicants present their research, the company, and then their protocols. So that will help expedite a lot of this so there's not additional redundancies occurring when we're talking about contracting or permitting or anything like that. So that will be how we'll do that. And then we'll have them finish each presentation and have a larger Q&A at the end. So if you have any questions, comments, or concerns, save them till the end. Make sure that you note which specific applicant you'll be referring to. And then we can ask those at the end to get anything that you might have concerns about onto the record. So I'm going to go ahead and hand it over to Don. Don, you should be listed as a co-host. If you want to present, you are free to do so.
Thank you, Colin. Thanks everyone for participating today. Let's see. So I think you're not seeing the right thing. How's that?
Good.
Looks like Tim's trying to get in.
I think Tim was admitted.
Okay.
Yeah.
So so today I'm going to be going over the facility information and my name's Don Crooks, by the way. I'm the senior EHS manager for Flagship Pioneering. I am representing these 5 companies as they are smaller companies, and I provide support, EHS support for them. I'll be going over general information today, the facility information, like I said, and then the safety information. And then once I've completed that, then we'll get into the individual companies and they can present on their science. So to start off, the facility is office and laboratory space located on the 4th floor of 101 South. And approximate square footage of the lab space is 17,000 feet. So for security, exterior building doors will always be locked. And for the company space, each, in order to get actually up the elevator, it's key card restricted. And visitors must sign in at the security desk. The lab design is for BSL2. And in the tissue culture rooms, we have separated tissue culture. So for each company that might be sharing a tissue culture room, it'll vary based on the company, but they will be designated BSCs for each of those companies to stay separate from each other. They won't be sharing BSCs. We also have BSL2 Plus enhanced PPE protocol for lentivirus work. And I'll get into that a little bit more. So for the lab space allocation on the 4th floor, with the exception of the yellow, which is just the office space, these are each of the companies that will be presenting today and their allocated space on the 4th floor. Some have more space than others. And then if anyone has a question about the shared space is typically shared equipment that will be stored in that area. So, types of equipment that's provided by Flagship that they may not necessarily have as a company. For infection control, employees at risk of occupational exposure to bloodborne pathogens are offered Hep B vax. and/or a titer at the time of hire. All lab employees receive bloodborne pathogen training within the first week of hire. Occupational health services are available through Mount Auburn. All of them have a Mount Auburn agreement. Emergency response and exposure control protocols are outlined in the biosafety manual and biosafety and exposure control plan, also within the emergency action plan. So for general biosafety, the main labs, as I had mentioned before, is BSL-2, shared tissue culture rooms, BSL-2 and BSL-2 plus. For PPE, button lab coat, safety glasses are required in the laboratory. Gloves are required for bench work. Recommended additional PPE for the tissue culture lab when dealing with like BSL-2 plus lentivirus includes splash goggles if splashes are likely, disposable lab coat over your regular lab coat, and disposable gloves. So for training, all new hires receive in-person and computer-based training during their first week of hire. That includes emergency evacuation, lab safety, chemical hygiene, biosafety, and bloodborne pathogen. And refresher training is provided annually. Training records are stored on both an online shared safety folder and a third-party platform. Some of the laboratory policies that we have, eating and drinking and applying cosmetics are not permitted in the lab. Laboratory guidance prohibits the handling of personal electronics while wearing gloves. And the glove policy is gloves are not allowed on laboratory door handles leading to the office space, and internal laboratory doors are deemed contaminated throughout, which means that they should be wearing gloves when they use the door handles. All waste and biological material must be completely sealed prior to leaving the lab. For tissue culture rooms, universal precautions used when handling human source material. It's treated as infectious. For waste disposal, liquid waste from cell cultures, chemical disinfection using 10% bleach for 30-minute contact time. For dry solid waste, tips, tubes, gloves, all solid biological waste is collected in specialized containers. Waste is removed by a licensed medical waste hauler. For sharps, they're all collected in rigid puncture-resistant containers and disposed of by lab support. Recapping of needles is not permitted. Waste storage is located on the first floor of the chemical storage room and is managed by Unity Lab Services. And our medical waste hauler is Triumvirate. So moving on to the permits and timeline. So for RDNA, we're obviously doing it right now. Wastewater, the landlord owns the MWRA permit. Flammable storage, hazardous material process, and hazardous material storage permits are all in progress currently. Once the labs are established, I believe we'll be getting a walkthrough. And then, excuse me, each of these companies are considered very small quantity generators of hazardous waste, and they're currently in the process of obtaining a EPA ID number for the particular site. Contracts in place, as I mentioned before, Mount Auburn Occupational Health Services, Triumvirate for biological waste and hazardous waste. Lab coats are provided by Cintas. BSC certifications is Air System Technologies and Triumvirate, and that's currently in progress. So once the BSCs are moved from Cambridge, they will be recertified. Fume hood certifications is provided by EH&E. Pest Control Services, Waltham. and our safety consultants are EH&E. And that was it. Any questions?
Thanks, Don. We'll save questions after all presentations are completed. But I guess my question is, who's up next? Who's presenting first?
Well, we can start in alphabetical order with a priori.
Awesome. And who should I make co-host?
That will be me, Sukhmani.
Excellent. All right. Sukhmani, you are officially a co-host. Congratulations. You're all set to present.
All right, just give me a second to share my screen. All right. All right, can everybody see my screen? Awesome.
Yes.
All right, hi, I'm Sukhmani Bedi. I'm a senior scientist with A Priori Bio, and I will be presenting to the Biosafety Committee on behalf of the whole company. Our scientific management includes our CSO, Harry Clintos, and we have two associate directors who are leading some of this management. So the mission of A Priori Bio is to develop improved vaccines through high throughput selection of viral entry proteins. We are building platforms to determine again, our platform is generalizable to a lot of viruses. And our idea is to build safe platforms to determine which viral entry proteins make the best vaccines. And the on-site use of human biospecimens, recombinant DNA, lentivirus, and an attenuated influenza virus is necessary for us to be able to do the science that we are proposing to do. And I will go into a little bit more details on what this really entails. In terms of the specimens or the agents that we are going to be dealing with, we do quite a lot of human sera or plasma screening. We work with immortalized human cell lines as well as primary human cells. Some of our display technologies for viral entry proteins involve yeast and mammalian cells. For our display of virus display technologies, we work with a second or third generation replication incompetent lentivirus system. And then just for seasonal influenza, we work with a lab-adapted non-human pathogen strain of influenza, which is the WSN strain, and this has been serially passaged in mice for about 50 years now. So it has basically lost its ability to infect humans, and hence it's not a human pathogen, but it is still replication competent in cell lines. If there are any questions on this slide, I'll be happy. Actually, we decided to take questions at the end, right? Yeah, okay. All right, so for any non-virus work, we follow the BSL-2 procedures, where personnel where we expect everybody to wear a lab coat, safety goggles, as well as gloves. We have designated area where we do certain workflows. So for any mammalian cell cultures or sera work, we work in the biosafety cabinet within our TC lab. And for any yeast work, that can be conducted in the main lab area. So our TC space is basically, it's a separate room from the rest of the lab. In terms of all of the waste that is generated during this work, it is all discarded in biohazard bins. Of course, the sharps are segregated from the rest of the waste. And then any potential spills will be decontaminated with 70% ethanol. In terms of any virus work, which is both lentivirus and our attenuated flu work, we as recommended by Flagship, we actually follow BSL-2 plus procedures, even though these agents are BSL-2 based on the CDC. On top of our usual lab coat, we do wear a waterproof a disposable waterproof lab coat that is discarded every time we use it. We also do double gloving, and of course, there are safety goggles. The work will be conducted in a designated room, which is the TC Suite 4301. And there is, of course, signage outside indicating what kind of work is being done. No virus leaves this room before inactivation, and the inactivation, depending on, could either be with just a decontamination with bleach, or for certain workflows, we do use paraformaldehyde for 30 minutes before we can take the samples out and do further processing if needed. For any waste that's generated, such as tips, serological pipettes, or plates, we first decontaminate them in a beaker inside the BSC for 30 minutes, and before we drain all of the bleach or the microchem, and then they can be discarded into the biohazard bins. The centrifugation is all done in sealed tubes, and these are only opened inside the BSC after centrifugation. In case there is, any accident, nobody gets exposed. And these buckets are sterilized with 70% ethanol before being taken out of the BSC. The virus stocks will be inventoried and stored securely in a designated freezer. There is no use of house vacuum when any virus work is being done. Everything is first, all of the liquid waste actually goes directly into a beaker with bleach or microchem, and we're allowed to sit for 30 minutes before it is dumped into the sink. And all personnel conducting any influenza work are required to demonstrate proof of influenza vaccination. And again, this is not because they're at risk of being infected with this system, but this is more for perception. And we just want to make sure that if there is an unrelated infection, we can make sure that we are protecting people and it cannot be traced back to the work we are doing. This is all I have. If there are any other questions, I am happy to answer, or I think we're taking questions at the end, so I keep forgetting that. Yeah.
Thank you, Sukhmani. I really appreciate it. Excellent presentation. Don, who's up next?
Let's go with FL 101. I think,
Yep, that's me, Bharati Sundaresh.
All right, and you are a co-host. There you go.
Great. Okay. So you'll see that there are some similarities to Sukhmani's presentation, but continue to share. Hopefully you can see the right screen here. So FL 101 is a small but mighty team.
You might want to click on swap displays.
Oh,
so
here, I'm just going to unplug my. There we go. Okay. So FL 101 is a small but mighty team. I stated here name pending. We just received a round of funding to essentially transition to our Series A round. So we have a team of 3 current employees, 2 of which are experimental, will be in the lab, and we anticipate growth to around 8. And our company mission is to profile cell-cell contact that we know is really a hallmark of disease, to build the world's leading contact biology company, to create a contact atlas through the technology that I'll speak a little bit to, that will feed into therapeutics that either disrupt cell-cell contacts or engage cell-cell contacts. And the team is managed by a president, CSO, myself, and a head of strategy. So FL 101, so going back to sort of this figure right here, I mentioned that we're profiling the contact between cells. So in a co-culture system, you can imagine when you have a cell one of a particular type and a cell two of another type, we're repurposing lentiviral machinery to track the contacts between these cells. So essentially, we're using lentivirus that is equipped with a fluorescence reporter and a barcode and turning, measuring contact into a sequencing output. So we're using recombinant DNA to study these cell-cell interactions. These viral nucleic acid sequences are either synthesized de novo or just sourced from a commercial vendor. And they encode just the standard lenti fusogens, replication factors, and these are all factors that have been previously studied in viral transmission. And just wanted to clarify too that this is third generation RISC-2, and any addition of an envelope or accessory proteins are characterized here. I've also listed the human genes that might function as viral restriction factors in some of our experiments. And I also mentioned that we include a recombinant nucleic acid fluorophore. So here I have mCherry GFP. We've also done another green fluorescence reporter, so the nucleic acid for that, to track the cell-cell contact by a flow cytometry readout, and also a barcode that tends to be 8 to 12 base pair in length. The cell lines and that we tend to work with are pretty standard. To date, they've really centered around HEC293Ts, K562s, DRCAT-Ts, branching into primary T cells now, and we anticipate further work in PBMCs from both healthy donors and those with disease conditions. And for any plasmid DNA production, we use E. coli, and it's at relatively small scale. So FL101 operates mostly at BSL-2 plus. In our lab at Boynton Yards, we will have a external lab space and lab benches, and also a dedicated TC space. All of the lenti work and cell culture work will be in that dedicated tissue culture space, while the exterior lab area is really for molecular biology and sequencing library prep. The lentiviral vectors will be handled at the BSL-2 plus protocol. I think a lot of these details were discussed earlier, but just to say, we'll take the universal precautions and the below safety measures while working with lenti. Waste disposal, again, is fairly standard. Liquid waste will undergo chemical disinfection. Dry solid waste will be collected and stored in the dedicated either waste room. Here it says first floor. I think that's from our previous location. Sharps will be in the appropriate sharps container, and BSL-2 plus waste will also be deconned before exiting the BSC.
It will still be on the first floor.
Okay, great. And a couple other details here on waste disposal. And I think a lot of these details were covered in Dawn's beginning presentation on our health and safety programs, but all employees, receive bloodborne pathogen training in the first week of hire, and additional training is provided for the employees working with lenti, which right now we have 2 experimental scientists, and they both do. And I think that's it on my end.
Thank you, Bharati. I really appreciate it. Excellent presentation. Don, who's up next?
Go with FL 107.
That would be me, Paris.
Paris, you're up, and you are co-host. Go ahead.
Excellent. Can you guys see my screen? Fantastic. All right, so thanks everyone for making the time. My name is Paris Palace. I'm an associate and part of the scientific management team at FL107. Okay, this is stuck. All right. And so together with Chaz, who was part of the other member of the scientific management team, we lead FL107, which also does not have quite a name quite yet. Our mission is to leverage novel biology and chemistry to control cellular proteostasis by leveraging what the proteasome does. And so what we really do is we're building a platform company that can use synthetic proteins, but also small chemical molecules that can influence protein homeostasis. And so we'll use nucleic acids or recombinant DNA to express these synthetic proteins and also to express the targets of these these synthetic proteins. And this will help us model or create cell culture models for target discovery and validation purposes. This research will happen at the location that Dawn introduced. And so all of the procedures we're doing, they're essentially the same ones presented previously, fall under the risk level 2. So we have a number of different cell lines. I think all of them were covered before. We also use this mouse cell line called BN3. It's an endothelial cell line that's not really infectious in any way or form. We're using third-generation lentiviral vectors for transductions, but probably are just going to do this a few times, leveraging FL101's capabilities and their space. And then we will transfect, as mentioned before, with recombinant nucleic acids encoding for human proteins. And so similar to what the previous presenters introduced, we will use these immortal cell lines, and they will come from mostly ATCC, and some of them will be generated by us via lentiviral transduction, and this is what we use the third-generation lentiviruses for. And then we'll use tissue culture to validate the expression activity of these other synthetic proteins that we're using. And so we'll use them to either create stable reporter cell lines, or we will add these constructs to express synthetic proteins and express them exogenously. These constructs will not be infectious or self-replicating or self-amplifying in any way or form. And all of the materials that we're using will be disinfected with ethanol bleach or a combination thereof. Lentiviral transduction was also discussed earlier, so we're essentially just using the exact same approach, but we'll use this to deliver different therapeutically relevant proteins such as alpha-synuclein or epithelial growth factor receptor. And these gene products will just be mRNAs that encode for the proteins mentioned above. And none of these are infectious or self-replicating. Similar for our plasmids and transfection, we'll get all of these by commonly known vendors such as Thermo Fisher or GenScript. These recombinant DNAs will just encode for an mRNA that expresses a synthetic protein of interest. And none of these are, again, either infectious nor self-replicating. This was covered before, so risk group 2, these are our third-generation lentiviruses. Research facility covered by Dawn. So we will be in the places indicated here. I also am highlighting here that we will use, for lentiviral work, we will use FL101 space, which is next to this red shared space. And we will share FL1, our TC space with Metaphor, which is another company neighboring. And our office space will be on the 6th floor. This is less relevant for this presentation. Infection control was covered by Dawn. Same for general biosafety. The policies were covered, waste disposal, and no rDNA work will be conducted prior to approval by the SPC. That's it. Thank you.
Thank you, Paris. I believe that leaves Metaphor up next. Is that correct, Don?
Yes, that's correct.
All right. Who is up for Metaphor?
I am.
Oh, hi, Susanna. Go ahead and wait. Yes. Wow. You should be all set, Susanna. Let me know.
I also want to apologize because I'm substituting for Casey. So this was a little bit of a last minute for me. So hopefully we can get through this very smoothly. Can you see my screen? Uh-oh. Can you see me?
Yeah, we can see you, Susanna. You had there a couple times in presentation mode, but your cursor was doing this weird
Okay.
pop-out thing. So
Let's see.
maybe try it again.
I'm going to share the desktop.
Yep.
Can you see it now?
See you now.
Okay. All right. So good news is the other companies have already covered a lot of the topics. So hopefully we can go through this pretty smoothly. About Metaphor, so we are also an early stage biotech company. We are probably the biggest of all the companies that have been presented today with around 50 employees. And we were also founded much earlier in 2022. We are in a place in between computational and experimental pipelines. And the idea or the mission is to really be able to design biological drugs at scale, really being able to cover a higher space of diversity and doing all of this in a much more accelerated timeline. As I mentioned, we are about 50 people. We just recently got a new CEO, Angela Wang. We have a total of 3 VPs and 5 directors and senior directors managing a team ranging from associate directors to research associates. And all the work that will be presented here is biosafety level 2. Very quickly, as how our experiments, the technicalities of our experiments, to put it into context. So as I mentioned, our goal is to identify and characterize therapeutic peptides and protein sequences. And the way we are doing this is, number one, by library screenings. And so we are using organisms like bacteria, phage, yeast, or mammalian cells to display protein and peptide libraries, and using screening methods like flow cytometry or binding or magnetic capture for selection. After we have identified our sequences, we produce them recombinantly, and that's where the DNA come in. And this production can happen either in bacteria, yeast, or mammalian cells. And here we are using very standard mammalian cells that other groups have already described. The step number 3 would be the characterization, where we apply a variety of molecular and cellular techniques to assess the properties of our peptides or proteins. And so here is where the BLS2 comes in, where we might be using some cell work to understand the function of our molecules. And primarily we are using immortalized cells, but sometimes we are also using primary cells like PBMCs that have been derived from animal or human blood products. Additional training is always necessary when users are working with human-derived products, and that's something that we are always very careful about. So, of course, as part of this characterization, sometimes we might need to have some custom systems where they are overexpressing a protein or a reporter system, and that's where the lentiviral work comes in. I have detailed here some of the safety measures that we are taking when working with cell culture. They have been already covered by the other companies, so I don't know if you want me to go one by one. We can always come back here if you have more questions. And same with the lentiviral work. A priori described it very nicely and very thoroughly. So we are really following the same exact safety measures with very thorough training of new users, as well as disposing of the waste. As for the sources of nucleic acids, so primarily we are using them to generate proteins of interest. These proteins can be of many different natures, from receptors on a surface to antibodies to signaling molecules. We do not work with toxins, and usually they can be either from human or from non-human origin. And that's it. I really apologize. I was told that I was supposed to be presenting with a very short notice. So I hope that this kind of covers a little bit the whole context of what Metaphor is trying to do.
Thank you, Susanna. Really appreciate it. And that leaves Prologue.
Last but not least, Prologue.
Perfect. All right, Prologue, you are co-hosts officially.
Okay, beautiful. All right. Thank you so much. As Dawn mentioned, last but definitely not the least, Sid and I will walk you through Prologue's mission and how we are prepared for our work at Somerville location. With that, Prologue launched in May 2024. We are currently 23 employees. In fact, this year we actually appointed Raj Chopra as our CSO. And as I said, like for this presentation, Sid and I will be walking through on both the ends on how we plan to use some recombinant DNA technology and lentiviral work with Prologue's mission. All right, so the main mission with Prologue Medicines is searching through the vast viral proteome to identify proteins for understanding whether they're useful for therapeutics. With that, again, as many other companies did point out, that the risk level 2 labs are very well ready for us at the Somerville location, but also we are preparing ourselves for Prologue Medicines in terms of the usage of our BSCs. We will have badge access, that's already been mentioned by Dawn. We will have certain signage as well outside of our BSCs to represent ongoing work inside, be it regular expressions versus lentiviral work. For lentiviral work, Sid will walk through, but we also will have enhanced PPEs, as mentioned by Sukhmani and a few others that will follow the guidelines as well in terms of when we do the lenti work. Other than that, all generated waste will be treated with 10% bleach for 30 minutes before disposal, and everything else that was, again, previously listed by other companies would be what Prologue would be following as well. And most importantly, annual refresher trainings are always given to everybody who works in the company, and obviously to all the new hires as well. With that, we have two aspects of the presentation today. One with general rDNA work. So basically, we use both human and viral secreted proteins that we express in a well-known expression vector like a BCDNA 3.4 that we then express in mammalian expression systems like HEC and CHO. We further then take these proteins for testing them for binding and activity using various assays, and then we finally use them for cell-based assays as well, which are mostly commercially bought cell lines. Just as a quick snapshot again for the plasmid production, especially, there's two ways we do this both in-house in 96 per plate or at a MIDI scale. We usually outsource it to GenScript and GeneVis for mega and giga preps. But outside of that, GenScript is our main CRO that helps make some of these plasmids in basically E. coli. We also have this in-house platform called the Telesis Bio that actually produces some of these genes for us in an overnight run. It's a custom build. Again, we have our vector that we always use, which is PCDN-4. As I mentioned, once we have these plasmids, we usually put them through expression systems like 293 or CHO, following which we produce them and purify them in small scale and large scale up until CMC grade proteins. Again, as I mentioned, for most of this work, we do have well-maintained lab and TC space to handle this work. And more importantly, we have trained personnel with more than 5 years of experience in handling plasmid and protein production work. With that, I'll pass it on to Sid to walk us through the lentiviral work.
Thanks, Shruti. So we use lentiviral work primarily to generate cell models for some of the work using the proteins that Shruti described. And I just want to say this is not something we do routinely. Lentiviral work is really intermittent. We may do some lentiviral work once every 2 months, if that. But we primarily use two approaches. One is to use CRISPR-Cas9 guide RNA libraries to basically knock down expression of certain genes of interest, or we basically use lentiviral transfection to overexpress certain genes. And the idea in both of these situations is to basically alter the genetic profile of the cell lines that we're using to basically make either overexpressors or knockouts. The cell lines that we use for this work are primarily immortalized cancer cell lines. So your examples of hex cells or JUCATs. So we don't really do this work on any cell line where we anticipate causing any unexpected phenotype. Next slide, please. A standard workflow, as some of the other groups have already described, is we basically have a cell line we transfuse with basically a state-of-the-art Gen 3 lenti construct. We don't packer lentivirus in-house. We buy lentiviral particles from a commercial vendor to reduce the risk of contamination and spillage further. Once we've transferred our cells using standard protocols, we basically start a selection. And once we have a positively selected clone, then the cells have stably integrated a construct of interest, and they can then move out of the lentiviral specific TC hood. The work that we do is supervised and is only performed by scientists who have experience working with lentivirus. So at present, there's basically 3 of us in the company, and with all the proper safety protocols in place that Don and other groups have already described. Thanks.
Thank you.
Thank you.
Thank you both for the presentation. Really appreciate the detail and all the information. So that concludes the individual applications. We can open it up to a Q&A for committee members and then from the public. If you have a question, there's a react button towards the bottom. Is it here? You can react, you can raise your hand, and I'll recognize folks in order of when they raise their hand on the screen. So, all right, I recognize Michael.
Thank you, Colin, and thank you to everybody who presented. Very nice presentation. I have a question for a priori. As far as I can see, you're the only ones who are using reproduction competent model, the attenuated flu, and you're adding, please correct me if I get this wrong, but you're going to add potential pathogen proteins to those attenuated flu. Is there any concern about those flu gaining function from the proteins that you're adding?
Yep, that's a really good question. So that's why any work that we are doing with replication competent flu is only going to involve sequences from natural circulating strains. So based on the guidelines from the CDC, we can work with sequences from the last 10 years. And because anybody coming into contact, even accidentally, has been vaccinated, or you would think they've been vaccinated. So we will not make any mutations that don't exist in nature or haven't existed in nature in the last 10 years.
Thank you. Great question, Michael. Thank you, Sukhmani. Francine?
I have a couple questions for Dawn. One, for the shared equipment that you had mentioned, those shared rooms, how are you ensuring that they're properly decontaminated between users, between companies?
Those are maintained. Well, first off, the company that uses them has a protocol to clean the machine after they're finished with it, and then we assure that with our lab manager and ULS, Unity Lab Services.
Okay. And then it could be just the way you had phrased it in your PowerPoint, but you had mentioned that the gloves had to be worn the entire time in, and you were considering door handles in the lab to be contaminated because gloves would be worn at all time. But does that also include the exit door out to the office spaces? Because that would.
No.
Okay.
No, yeah, the door leading to the office space, you're not supposed to wear gloves.
Okay. Just the way it was presented, it seemed that you were requiring it, which was counterintuitive.
I'll have to check my wording.
Wait, I did actually have one more question. I guess it could be for any of them since all of the groups mentioned it. But using like a bleach solution sitting in a beaker in a biosafety cabinet can lead to potentials of that being knocked over and then spills. Have any of you considered an alternative of using, a small bag with some paper towel in it in the back of the cabinet as opposed to using the bleach in a flask or beaker specifically? Any wrong?
Can you repeat that? What was the alternative?
It's typically to put like a smaller bag, whatever the max length is for your serological pipettes, but you actually have it sitting in the back of the cabinet. It makes it easier to pull a serological pipette and slide it in to the back without crossing over or potentially knocking over a beaker with bleach in it. I do biosafety normally, so
We hadn't. I don't think
potential
we've
for knocking
considered
over
that.
knocking over the beaker that contains bleach in it.
Yeah.
And then you will bag up you'll close up that bag so that you are taking out something that's closed, and then you just need to wipe off the bag a little bit with the solution before, and then just take that out to put it into, since the final destination for it was going into your biological waste as mentioned, but it reduces that potential for knocking over the beaker. Just a thought.
It's a good point.
Thank you, Francine. Bill?
It was nice to meet all you folks, being the first meeting. Really appreciate a lot of it's weird on my head. I'm going to go to the area I do understand, the emergency action. So first thing I want to do is, in this, most of my stuff is for Don, because he kind of has the overarching. See, in the plan, it says, in the case of an emergency, on page 12, says in case of an emergency goes beyond property boundaries, the EC will notify Somerville Fire Department, which serves as the LEPC. I think I said that earlier. I just wanted to flag that we now have a formal local emergency planning committee. So if we could maybe update the plan to, go to the LEPC versus the fire department, which was serving as that. So I wanted to flag that first and foremost. The next question I have is when it comes to access and functional needs and evacuations. And so you guys do address it in your EAP. The one paragraph I saw that did kind of address it says responsibilities of the fire wardens include identifying and informing the EC of all disabled individuals, including temporary disabled, in the assigned area, assigning capable department representatives to aid in their evacuation, and identifying evacuation procedures. Now that basically identifies that you see that there may be some assistance needed and you've identified people to do it. But my question is, how are you physically doing that? There's no elevators available if someone's in a wheelchair. That's something of the staff able to help someone leave. And if not, have you implemented any other procedures to assist in those areas?
So we have fire wardens assigned, or they volunteer to be fire wardens, and we have training associated with it. And within the training, it's stated that if you have somebody So the key to this is knowing beforehand, right?
Mhm.
And and to risk assess how to get that person out before the emergency happens. So if a fire warden knows of somebody who's disabled within their scope, they should be reaching out to myself or somebody or the lab manager to to take a look at it and determine how best to get the person out of the building.
Yeah. One of the things I look for in the plans is like if they can't physically get the person out, identifying or designating on your evacuation map something like an evacuation assistance area. So then the fire department knows if people can't evacuate on your map, it says like maybe like near a stairway, this is where people will go. And this is where we first look for them if they can't evacuate on their own with assistance. So that's just a recommendation I have on my end, honestly, for best practices. But I thank you for that. It actually, the plan was very good. I found areas I was like, oh, I want to find out about this. And I kept looking up. There it is. There it is. So very comprehensive plan. So the last thing I have is in the emergency drill schedule. The plan says the EC will schedule and administer periodic fire drills. How do you define periodic?
Well, periodic is typically once a year.
Fair enough. And you guys have language, and if you have a real life incident or even a false alarm, you can analyze that and say, we did this well, that well. So I appreciate that. So I just wanted to delve into that because it's kind of a broad term. So I appreciate it. Thank you.
Yeah, thank you for the recommendation, though. We'll definitely look into that.
Thank you.
Thank you, Bill. Any other questions, comments, concerns? I have one quick thing that I noticed on the certificate of insurance. It doesn't have the city of Somerville listed. I was wondering if we can get an updated certificate of insurance with the city also added on there. That would be great.
Sure. I didn't realize that it didn't have Somerville listed.
Yeah, it's also a very small little section and then the 13 different requirements we have for this. So it's often missed. So just wanted to flag that for everyone.
Okay.
Any other question? Oh, Francine, you're back up. Go ahead.
So yes, so this would be more on Don because everybody said it correctly in their presentations, but agents have risk groups and practices containment are biosafety levels, and in the form that you have everybody filling out, it was like, and partly in section, I think it was D3, and then there was another section in an earlier page 6 for APR8. You have it listed as being BSL2 agents. But agents have risk groups. And I will say everybody's presentation, their PowerPoints, they listed things as being risk groups too, but your registration form implies biosafety level 2 for agents. So it's really small, but
you're just saying that we should spell out the risk group along with
you use the term risk group instead of biosafety level 2 when you're talking specifically about the agents.
Oh, I see. Okay, yeah.
The containment, the facilities are BL2 or BSL2, but the agents themselves would be listed under risk groups. And I have to say, everybody's presentations like listed them as risk groups, I noticed, but your registration form that you're having them fill out said biosafety level.
Okay, thank you.
Thank you, Francine. Anyone else have any more questions, comments, concerns? I see we have some folks from the public here as well. Anyone from the public have any questions? All right. Seeing none, I'm going to motion for approval and completion on conditions that some of those minor details are altered and submitted through the portal in a timely manner before payment is received. Do I have a second?
I second.
Great, excellent. Couple seconds. So I'm going to call the vote for approval on condition that corrections are made that were discussed during this meeting and be reflected in the minutes. Let's see. Bill Fisher,
Aye.
Alex Vera,
I'll have to abstain. Thank you.
Michael Swickus, Francine Rogers,
Aye.
Deputy Chief Bill Hallahan,
Aye.
Elise Belarge,
Aye.
Karen Carroll, absent. Colin Ziegler, aye. And with that, the approval passes on the conditions that those corrections be made and submitted through the CitizenServe portal before payment is received. So thank you all for your presentations tonight. I know that was quite the meeting with a lot of information. If the public or any other folks in attendance have any more questions, comments, or concerns, we're happy to field those. Again, Don will be in contact about the approval with conditions process and then payment as well. So thank you all for your excellent presentations and the information. It was great to see you all and learn a little bit about what you're doing in our backyards. So it's very exciting. Thank you. All right.
Thank you.
Yep. Thank you. Last item.
Thank you.
Last item for tonight that we're going to move to is an update on the renewal period. So we finally figured it out in CitizenServe. All previous permits that were going to be cycled through this previous one, we're going to be giving a payment pass for. The renewal did have to take place, of course, but they're going to be cycled into a January 1st through March 30th renewal period, which will help this committee with scheduling and get a better idea of when we're going to have a bulk of renewal reviews rather than doing them periodically throughout the year. So that is exciting on our end. Those who hold those permits have already been notified and have received an updated license as a result. So that is pretty much it. Any other questions, comments, concerns from the committee members this evening? All right. I'm going to motion to conclude this meeting. And do I see a second? All right, I see some seconds. All right, Bill Fisher. Michael Swickus.
Aye.
Alex Vera.
Aye.
Elise Belarge.
Aye.
Francine Rogers.
Aye.
Am I missing anyone else? Colin Ziegler, myself. Aye. Karen Carroll. Absent. And then Rachel Ned Carney was here temporarily, but she had to leave. So I will forward any questions, comments, or concerns onto the applicants if she had any. Thank you all again for your time this evening. Feel free to reach out if you have any other comments on how tonight went. So thank you.
Thank you, Colin.
Thank you.
Thank you, everyone. Have a good night.